This video is my interpretation of the second half of chapter 18 on cell division. Mitosis, the nuclear division phase, can be viewed as a stage in which the chromosomes are performing a dance, so that each daughter cell gets exactly half the chromosomes from DNA duplication. M phase, mitosis phase, represents a small section of the entire cell cycle. But in this figure, we have just enlarged that section to get the words in their appropriate places. So students need to learn that prophase is the first phase of mitosis, followed by prometaphase, metaphase, anaphase, and telophase. Cytokinesis is not part of the actual nuclear division that takes place. It is an extra phase which sometimes overlaps with telophase that results in the production of two daughter cells. If students could learn at least two facts for each stage of mitosis, that should help them in subsequent analysis and assessments. In a cell cycle, all other phases, G1, S, G2, and even G0 are considered to be part of interphase, so they're not part of the M phase. However, events in interphase do prepare the cell to perform M phase, mitosis phase. So during interphase, the DNA is replicated during S phase, and other organelles are duplicated too, so that they can be shared evenly amongst the daughter cells. One of the notable conditions that takes place in late interphase is the duplication of the centrosomes, the structures from which microtubules will originate and perform a major task during M phase. The actual mitosis phase begins with prophase. In prophase, for the very first time, the chromosomes begin to condense, and they can be seen under the light microscope as individual bodies. The nucleus, however, is still intact. But in the cytoplasm, the mitotic spindle is beginning to take shape, as that will be necessary in the next phase of mitosis. In prometaphase, which is sometimes left out of books, some sequence of events takes place that is very important. The nuclear membrane begins to fragment and disappear into the background. The chromosomes are now so short and fat that can be seen as individual entities. And each chromosome, as the nuclear envelope collapses, each chromosome is captured by the unseen microtubules of the spindle system. Metaphase is perhaps one of the shortest phases of mitosis, where the chromosomes are pushed and pulled and arranged in the center of the cell in a 3D arrangement that's known as a metaphase plate. You can see it in the actual figure here, that the chromosome centers are all sitting along a single dimension, along the middle of the cell. Their tails may be flopping to either side, but their centromeres are all positioned in the middle of the spindle. Once everything checks out in metaphase, anaphase quickly takes place, where the individual sister chromatids are separated from each other, and now they become known as chromosomes in their own rights, are pulled towards the different poles of the cell. The final phase of mitosis is the arrival of the chromosomes at the poles of the spindle and the reformation of the nuclear envelope around each pole. Each individual chromosome begins to lengthen and disappear. In most animal cells, the center of the mother cell begins to constrict at the contractile ring in preparation for the next phase, cytokinesis. Cytokinesis is not a phase of nuclear division, but it is a phase of mitosis. So here the constriction between the two halves of the cell continues until they pinch off, resulting in two daughter cells. The nucleus by then has become present, and the chromosomes within are no longer discernible as individual objects. What we'll do next is look at details within the phase of mitosis. So one of the major key events in mitosis is the actual separation of the sister chromatids. So it's important for students to know that the terminology is very, very strict. When chromosomes are still attached to each other at their centromeres, they are called chromosomes. So these two structures is a single chromosome. And they are known as sister chromatids because each one is a duplicated double helix. But as soon as the centromeres break up at the beginning of anaphase as such, they then are known as chromosomes in their own right. So this chromosome is now moving to the left, and this chromosome is now moving to the right within the same cell. This process is driven by the green lines, and the green lines represent microtubules and their associated motor proteins. And the process here is very similar to what we learned in a previous chapter, where the plus end of the microtubule begins to be degraded, resulted in the shortening and the movement of the chromosomes to their locations. Let's take a few seconds and study the centrosomes. As we know, the centrosome is a structure that originates microtubules. So it's a microtubule organizing center. One microtubule by itself is not enough to complete mitosis. So during an early point in the cell cycle, preferably between S and G2 phase, the centrosomes are duplicated and held in place. At the beginning of mitosis, the two duplicates begin to move apart in prophase, and by the time the cell reaches prometaphase, the two centrosomes have reached opposite sides of the same cell, and they establish between them a spindle. And the spindle has to exist prior to the nuclear envelope disappearing, otherwise the chromosomes would get lost within the space of the cell. This also, the loss of the nuclear envelope also increases the amount of space that the chromosomes have to do their manipulation. With this slide, we introduced the students to the word aster. An aster is a radiating structure that looks like a star. And in this case, each centrosome and its associated microtubules look like a star within the cytoplasm of the cell. Through dynamic instability, the microtubules begin to grow and collapse, as we saw in a previous chapter. They only become stabilized when they do two things. They either make contact with each other from the two opposite asters, or they make contact with the plasma membrane. And in this example here, in the center of this figure, the microtubules originating from the left and the right side, there's two sets here, have made contact. And that, in addition with motor proteins, stabilizes their structure and prevents them from collapsing further. What's not shown are those microtubules which are stabilized by making contact with the plasma membrane. That will be on this side and over here on this side. The primary function of the spindle is to control the movement of the chromosomes. Illustrated in this slide, we have a single chromosome with its two sister chromatids. One thing that forms during late prophase is the deposition of proteins on one side of the centromere, on every sister chromatid that's present inside the cell. So once these structures form, they are known as a kinetochore. It's a pretty sophisticated protein matrix that forms. The purpose of the kinetochore is to attach the microtubules of the spindle. As indicated here, in large animals like us, we have up to 40 different microtubules attached to each kinetochore. In other creatures like yeast, they sometimes have much fewer, such as one. The purpose of these green lines is to then control the positioning of the chromosomes within the space of the cell. Through basic research, we've accumulated a lot of knowledge about the structure of kinetochores as illustrated in this slide. Various proteins are involved in setting up the system along with many filaments. Once a spindle is fully formed, scientists can identify 3 different classes of microtubules as indicated here. So the first type are those microtubules that are just forming the aster. they undergo dynamic instability constantly. So some of these are growing and collapsing at the same time. But they are important in allowing the spindle to have some kind of base, a foundation. Then we have those microtubules which are attached to chromosomes, indicated in blue. And finally, we have those microtubules which connect one aster to its partner, and those are indicated in pink. And the whole system works in collaboration to perform the manipulations necessary to divide the chromosomes into two halves. It should be mentioned that centrosomes are not paramount in allowing mitosis to ensue. Indeed, some animals and all plants have no centrosomes. So studies like this have shown that centrosomes are not important and that the spindle can be established de novo inside the cell based on the existence of kinetochores. So the kinetochores of the chromosomes can drive the formation of spindle themselves. And that's what happens in plant cells and some insect cells. One of the most fascinating movements which takes place in mitosis is the movement of the chromosomes to the center of the cell. And that is during the phase of metaphase. And once the chromosomes are located there, then a check is performed before the next phase, anaphase, continues. This region where the chromosomes are lined up is called the metaphase plate. If all is well within the cell, then the chromosomes are permitted to enter the next phase, which is anaphase. So this is a very important control step in mitosis. Shown here are two photographs taken of two plant cells, where the chromosomes are stained in purple and the microtubules of the spindle are stained in red. And once again, there are no centrosomes in plant cells, but we can still see that there is some type of spindle that's organized into a two bipolar system. Let us focus on this transition between metaphase and anaphase. It's a very important step. We've learned in the past that during S phase, the DNA of chromosomes is copied. So here we can see a replication fork traveling down the chromosome to form two sister chromatids. And by the time the S phase finishes, we have two sister chromatids, but they're held together by these rings of cohesin proteins. So that happens by the time G2 begins. All during G2, the chromatids are held together to prevent them from getting lost or breaking. It's only during metaphase that these rings begin to be dismantled. And the entire set is not dismantled around the centromere until everything checks out. So once all the checks take place and the cyclin APC is building up, only then will these last few cohesin rings be dismantled, freeing up the two centromeres from going to opposite poles. We now have a pretty good handle on how things interact at this point. So the APC, the cyclin that drives this process, once everything checks out and the APC has been given permission, it then phosphorylates and also adds ubiquitin to a number of different proteins, including a protein called securin. And securin is an inhibitor of an enzyme called separase. And it's the separase which digests the last few rings holding together the chromatids. So the separase is necessary for completing the breakdown of cohesin, therefore releasing the two sister chromatids from each other. So here is another perspective on the same mechanism. So we have two sister chromatids indicated by the gray bars being held in place by the cohesin. And the cohesin has to be now cut open, and that's done by this enzyme here called separase. But separase is prevented from having an active site by being secured by securin. So the APC, one of its jobs is to add ubiquitin to securin. The presence of ubiquitin drives securin to be destroyed in the proteasome, therefore releasing separase to go and cut the cohesin. Once a cell is driven into anaphase, scientists have identified two purposes within anaphase, and they call those anaphase A and anaphase B. So on this slide, we're looking at anaphase A, and that's just focusing right here in the center. So the purpose of anaphase A is to separate the chromosomes from each other. And that's done by tension building up in the microtubules. And as soon as the cohesin, the last of the cohesin, is released, that tension forces the two chromatids to separate from each other, at which point they're known as chromosomes. And the chromosomes are then pulled towards each pole. In the second type of anaphase stage, anaphase B, which happens simultaneously, we focus our attention on the regions not involving the chromosome. So that would be the microtubules connecting the two asters together, and the microtubules connecting the poles of the aster, the centrosomes, to the plasma membrane, not shown. So both those systems begin to use motor proteins to drive the distance between the two asters further and further apart. So the poles of the spindle are forced further apart, therefore elongating the cell slightly. Why? To give the chromosomes a better opportunity to be segregated, to be separated. The authors of the textbook make a special mention of this spindle assembly checkpoint as a control system for driving the cell into anaphase. If all the chromosomes are connected to kinetochores, then a green light is given to the cyclin. If some of the chromosomes are missing their connections to the kinetochores and they're not lined up in the center at the metaphase plate, then APC is given a red light and the system comes to a standstill in order to fix that problem. And that's called the spindle assembly checkpoint for the onset of anaphase. Let's turn our attention to the nuclear membrane, the nuclear envelope as it's called by the authors in this section of the book. Here we see an electron micrograph of the inner surface of a nucleus, and we can see the nuclear lamina fibers as they're arranged in a crisscross pattern. And these stabilize the structure of the membrane of the nucleus, the double membrane. In this slide here, we can see the full sequence of events which takes place during each cell cycle. So here we have an intact nucleus, and that exists during G1, S, and G2, and also during G0. But when the cell is forced to perform a cell cycle, the migration of the nucleus into prometaphase results in phosphorylation of the nuclear lamins. So the nuclear lamins, the proteins on the inside of the membrane, are phosphorylated as well as the nuclear pores, and that causes them to dismantle. And the dismantling process allows the nuclear envelope to be then hidden inside the cell. So basically it dissolves away. Then later on in telophase, when it's time for the nuclear membranes to reappear, both of them, they do so by wrapping themselves around each chromosome and then coalescing with each other at each pole of the spindle, therefore regenerating for the daughter cell a complete intact nuclear membrane. This is a very fascinating way in which nature solves this problem. In animal cells, which do not have a cell wall, the plasma membrane is quite flexible. And during cytokinesis, the final phase of M, the mother cell is then divided down the middle into two daughter cells. And that takes place with something called a cleavage furrow. A furrow is an indentation. And that on the inside is regulated by a contractile ring. And the contractile ring consists of actin and myosin interacting with themselves, as we learned in the previous chapter, and forming a belt which tightens and tightens until the two halves are split into two daughter cells. How does the contractile ring know where to form and where the equator of the cell is? Well, as the spindle fibers begin to break down, the microtubules, they leave behind a small remnant between the two poles, and that remnant is used to locate the center of the cell, and that dictates where the cleavage furrow will form and the deposition of the actin and the myosin. One interesting and logical explanation that emerges from studying animal cells is that they change shape during mitosis. So here we have cells in interphase. These fibroblasts are flat and elongated, and they're stuck to the substratum, the surface on which they're growing. In this case, it should be a glass slide. But during mitosis, they round up and become spherical. Therefore, they must have let go of the contact between themselves and the glass slide. But after cell division, when you have two daughter cells, each daughter cell reestablishes those contacts with the substratum. This behavior is achieved by weakening the interactions between the cell membrane and the environmental glass slide. And that's done by the phosphorylation of proteins called integrins. We came across integrins in Bio 2 and earlier on in this course. These are proteins in the membrane which allow interaction across that membrane. So when these proteins are phosphorylated, they tend to be less sticky, therefore allowing the cell to round up. It also allows these cells to let go of their neighboring cells in tissue so that the two daughter cells can re-establish new contacts with their neighbors rather than relying on the neighbors that their mother cell had. Please learn that in cells that have a cell wall, it's impossible to bend the middle of the cell to form a cleavage furrow. So the solution in those organisms is to develop a structure that transverses the two daughter cells. So here we have a mother cell in a plant, and after nuclear division, the mother cell is divided into two daughter cells by the formation, bit by bit, of this walled structure called a phragmoblast. And it's made of fragments of the Golgi-derived vesicles that carry appropriate chemistry to re-establish between the two halves a new fresh cell wall. And that cell wall will then merge with the existing cell wall and partition off the two daughter cells. And then later on, the newly made phragmoblast cell wall will be then strengthened with the deposition of cellulose. Finally, we need to address what happens to the various organelles within the mother cell. And there are some solutions that are interesting. For most organelles, such as ribosomes, they are replicated early in the cell cycle, normally G1 and G2, and there are so many of them that by their sheer presence, it's acceptable to understand that they'll be randomly distributed evenly when the cytokinesis phase takes place. But for other organelles, there's a more directed outcome. Mitochondria and chloroplasts, they divide by binary fission, the same way that bacteria, their ancestors, divided. So mitochondria have to be informed to replicate themselves. So they grow in size and then they split down the middle. Now you have twice as many mitochondria as at the beginning. And the same thing happens with the chloroplasts. And because of their random organization, it's thought that their numbers would automatically encourage at least some to be present in each daughter cell. And if a daughter cell gets few, it can easily increase the number by undergoing binary fusion after cell division. So that's not a problem. The Golgi has an interesting life in that it is attached in fragments to the microtubules. And it uses motor proteins to migrate along those microtubules. And as the microtubules are distributed amongst the two daughter cells, they automatically carry with them this Golgi segment. So that takes care of the Golgi. And the ER, by contrast, is simply dislodged from the cytoskeleton and it floats around within the cell. Because there's so much ER, the chances of it being cut in half are increased by the cytokinesis process. So the ER is not handled in any way other than by volume. So the result is that the two daughter cells will have their own complement of organelles. Let's turn our attention to the final section of the chapter. So here we want to address how in a multicellular creature like us, we manage the number of cells and the size of those cells. The main process that we're going to learn about is apoptosis. Apoptosis is a very important way in which cells can be terminated, in that they can commit suicide by activating a biochemical pathway. During embryonic development, this process is more prevalent than you think. About half the cells in some places are resculptured by being apoptosed. The classic example you'll see later on is the development of fingers and toes. But at the same time, the process doesn't stop in adults. In adults, there's a continuing rate of loss of cells through various activities. We have the immune system constantly eradicating cells that are too dangerous to release into the circulation. We have repair mechanisms which are constantly removing cells that are old and need to be replaced. The process of apoptosis takes the dying cell through a sequence of physiological changes that can be seen under the microscope. So here we have a normal cell at the initiation of apoptosis, and we see the same cell a few minutes later performing this blebbing, where the plasma membrane breaks up into blebs and the membrane is turned inside out in the process. So the blebbing is mentioned here. And after that, as the parts of the cell fall apart, other cells come along and they consume through phagocytosis these blebs. So the blebs basically cut the cell into small pieces which can then be eaten by other cells. Returning quickly to the developmental use of apoptosis, we can see here in a limb bud how the bud produces a pad, and the cells that are marked in these little green circles will be encouraged to undergo apoptosis, generating fingers or toes. And the same thing happens when the tail of a tadpole is no longer needed. A large part of that tail will undergo apoptosis and be then lost in that fashion. Just to reiterate that there are two types of cell death. One is called necrosis, which happens when a cell is infected by a virus or when there's physical damage like an injury that takes place, versus the apoptotic system, which is considered a clean death. So here we have a cell that's burst open for some reason, and you can see it's spilling its contents into the environment. And that can cause inflammation and many other problems for that organism. Whereas in panel B, we have a cell undergoing apoptosis, and it's about to start performing blebbing, but before it does that, it has some biochemical changes that are taking place internally. And one of those changes is that the membrane gets turned inside out. And that makes that a target for other cells to come along. As you can see here, this is the cytoplasm of a macrophage, and then the cell is consumed by phagocytosis. So this will be considered a clean death because there's no spillage of contents into the environment. How are decisions made as to enter apoptosis, and what drives apoptosis? Well, the answer is the same as before. It's the chemistry of the cell. Biochemical pathways involving enzymes, phosphorylation, dephosphorylation, concentrations, and cascades are all responsible for driving these processes. There's more than one pathway that leads to cell death, and they can be activated using various systems. Some of these systems are internal to the cell when something goes wrong. Others are driven by cells or by external factors outside this one cell. Regardless, the main process at work is the activation of enzymes. In this case, we have a particular enzyme that exists inside the cell in an inactive form. Under a particular stimulus, the enzyme will be converted from the procaspase into the caspase version. So caspases are one type of enzyme involved in the apoptotic pathway. The cleavage of the heads removes the prodomains, exposing the active sites which come together in this dimer to activate something else. So the active site is formed down here when the dimers come together in this configuration. Once the trigger activates the initiator caspase, that caspase, amongst other activities, can drive the conversion of more caspases. Those are called executioner caspases. So students need to learn initiator caspase and executioner caspase. The executioner caspases have multiple actions. They can cause something to happen inside the cell, as well as driving the conversion of more molecules into the pathway for apoptosis. In this example here, these blue caspases activate these red caspases, and the red caspases may activate further caspases. And the red caspases have a different function to perform compared to the blue caspases. And this process will continue until all the systems are activated that cause cell death. It's important to note that once the system is activated, it's a one-way street. It's very hard then to stop anything from going backwards. Let us focus on one particular apoptotic pathway. In this case, the stimulus, whatever that may be, tells the cell to release cytochrome C. Cytochrome C is a molecule that's normally sequestered inside mitochondria between the two membranes. It's not allowed to leave because it's too large to pass through the outer mitochondrial membrane. But if it does enter the cytosol, it acts as a poison. The release of cytochrome C is under the control of two proteins called BAX and BAC. And those proteins can open channels that can release cytochrome C into the cytosol. Once cytochrome C enters the cytosol, it binds to always present adaptin proteins. And these adapter proteins then form a structure that self-assembles into this multi-headed protein complex. The protein complex then activates itself and attacks also always present this procaspase 9. And the two interact to form this structure here called the apoptosome. The apoptosome is then able to initiate a cascade of other caspases to be activated. So this is the key step, is the release of cytochrome C in this case. Once the cytochrome C is released into the cytosol, that is a one-way system. That is an intracellular death signal. It can't be reversed, and what happens subsequently cannot be stopped. Now, BAX and BAC are part of a family of proteins called the BCL2 family. Some of these proteins from this family activate apoptosis, and as we'll see in a moment, some proteins that belong to this family actually prevent cell death. So it's the balance of the two that determines whether a cell will survive or if the cell will die. The survival member of the BCL2 family is BCL2 itself. That's the name of the protein. When levels of BCL2 are high inside the cell, that protein is able to block apoptosis. Every day, cells in your body have to receive survival factors that bind to receptors on their surface, and through a process of signal transduction, transcription regulators are activated in the cytosol, which then are carried through the nuclear pore and act as transcription factors. And once they activate this gene, BCL2, the levels of BCL2 protein in the cytosol increase, and they can then block the negative effects of the BCL2 family. This figure here shows us a very rudimentary interaction that takes place between a particular set of proteins. The important concept is that the left-hand side of the slide here is the default situation. So this is what exists, say, in a cell at all times. And this system will lead to cell death. However, if survival signals are received by the cell and through a process of signal transduction, this protein AKT is activated through phosphorylation. The outcome of that sequence is to phosphorylate the protein bad, therefore releasing active BCL2. Then BCL2 will have some kind of activity which leads to the prevention of apoptosis. But this is a transient signal. The signal will have to be repeated on a regular basis to keep active BCL2 in this configuration and thus prevent cell death. If this signal is prevented from reaching the cell, then the consequences are the left-hand side and the cell will commit suicide. There's another system that cells have that does not rely on an endocrine or a paracrine signal. This system relies on a juxtacrine signal. So cell contact needs to be made. In this example here, we have a white blood cell, a killer lymphocyte, such as a cytotoxic T cell, a CD8 T cell. And on the surface of that T cell, we have a protein called the Fas ligand. And that will bind upon contact to receptors on the target cell. And they're called the Fas death receptors. And the binding of these two, as indicated in the middle panel, will cause the internal surface of the receptor to change shape. It will then be able to bind for the first time to this adapter protein, as indicated here. And once the adapter protein has changed shape, it will then be able to bind to its procaspase. And in this case, it's called procaspase 8. And binding to procaspase 8 will cause the procaspase 8 to become a caspase 8, indicated here. And that will then initiate a set of executioner caspases and lead to cell death. Interestingly though, once contact has been made, then the signal has begun, the T cell is able to leave and do its job elsewhere, and the cascade will continue. And within a few hours, the cell will have committed suicide. If the default pathway is cell death, then there must be mechanisms to prevent that on a regular basis. And we call those signals survivor proteins or survival proteins. And they fall into three major categories. First, as we've seen a few moments ago, are called survival proteins or survival factors. And when they bind to cells, they suppress apoptosis for a period of time. But they have to be constantly arriving on a regular basis, say once a day. Then an alternative way that cells can survive and perform cell division is through mitogen signals or mitogenic factors. And they are normally responsible for keeping the cell alive and overcoming any intracellular mechanisms that stop the cell cycle. And then finally, we have growth factors. And these growth factors stimulate an increase in the size of the cell in terms of its mass. And they do so by encouraging biosynthetic processes, these are anabolic processes, and suppressing catabolic processes when it comes to proteins and other macromolecules. So the sum here would be an increase in those molecules that would automatically increase the size of the cell. A great example of survival factors has to do with development and neurons. So the body has a habit of producing lots of neurons, extra neurons, and then allowing them to find their way towards the target location, like a muscle in your finger. And it's probable that a large number of neurons would find that pathway just by chance. but you only need one neuron to control that muscle. Therefore, the extra neurons are not needed, and ideally there's not enough space inside your arm for the extra ones. So how do you decide how many to keep? Well, the target cells are producing just enough of a survival factor, and the cell that gets the most dose will survive, and the others will commit suicide. Therefore, you end up with one muscle fiber being impregnated by one neuron. Mitogens, the chemicals, the proteins that promote cell division, are secreted by other cells in the body, normally neighboring cells, either in the same tissue or within important glands or regions of the body. And they activate cell division by an intracellular pathway mechanism. So once they bind to the surface receptors or to internal receptors, then a cascade, signal transduction cascade, ensues. And the way they work is to remove molecular blocks on the G1 to S phase. Examples include platelet-derived growth factor and hepatocyte growth factors. And these are used to govern the repair of biological pathways in the blood and, of course, to generate more liver cells. Similarly, we can predict how growth factors will work. They work very similar to mitogens. In this case, these proteins will bind to surface receptors through a signal transduction pathway. They will have two outcomes. One is to increase protein synthesis, and the other is to inhibit protein degradation. Therefore, the result is more proteins inside the cell and an increase in cell size. And you can see over here that different cells in the body, they have different architectures and different sizes, and different growth factors will impact these cells in a different fashion. Finally, just to mention that there are proteins in the environment which impact cells in the opposite fashion. They can inhibit cell survival, they can inhibit cell division, and they can also inhibit cell growth. So the great example of cell growth is indicated by this protein called myostatin. So myostatin is released in a certain dose inside mammals and most animals, and what it does, it prevents overexpression, oversized cells from being produced. How do we know this? Because if you mutate or otherwise remove myostatin, you end up with monsters like this that have an overdose of certain tissues. So these can be called hulky mice. The same thing happens in cattle if the same gene is altered there. And as the story says in the chapter, Belgian cattle were accidentally bred, and they happen to have a mutation in their myostatin gene. Finally, for those of you who have stayed with this video, here's a clue to what could be on the final exam. And I mentioned this in lecture, so people are not really missing out. So this graph here is very important. It actually shows you the amount of DNA inside sets of cells and how that DNA can change during the cell cycle. So the cell cycle has four basic phases: G1, S, G2, and M. And if we monitor the cells that are in different phases of the cell cycle, we can record the abundance of DNA on this relative scale. So this is the number of cells in that tissue that were examined, and this is the amount of DNA that they possess. One way to record this data is to label the DNA with some chemicals and then measure the content of DNA in terms of fluorescence as the cells flow through a flow cytometer. A flow cytometer is simply a hose that shoots cells through single file so that each cell can be analyzed as it drips from the top of the machine. And over a sample of cells, you get this graph here. So the interpretation of this is straightforward, that the amount of DNA should stay constant at one dose up to G1. And between G1 and end of S phase, the amount of DNA will begin to increase towards double the size. However, at the end of S phase, all the cells should have double the amount of DNA until they enter M phase. And here's M phase expanded. And the question is, how long before the amount of DNA drops back down to the original 1 dose? And the answer is quite simple, that it happens when the cells divide. So relating this graph to the cell cycle should be now straightforward. So this peak here at the lower abundance corresponds to the time when the daughter cells form up until the beginning of S phase. This plateau here corresponds to those cells which are in S phase. And as they go through S phase, more and more of their DNA has been doubled until they get to the end of S phase, when they're now at the second peak. They'll stay in the second peak through G2 because nothing happens, through the beginning of prophase, prometaphase, metaphase, and through anaphase. And then finally in telophase, when the cells start forming the cleavage furrow and actually divide, then they'll drop back down, each of those daughter cells, to 1 dose. Thank you so much.